Hi, I'm Megan.
And I'm Chloe, and we're going to be talking about the article Plasma-Soluble Prion Protein, a potential biomarker for sport-related concussions, a pilot study. So we wanted to start with this big question that kind of encompasses the entire paper that we looked at. So the question is, are these plasma-soluble cellular prion proteins applicable and a reliable biomarker for sport-related concussions that we're seeing so often today?
So first we're just going to get started by talking a little bit about a concussion and what it entails. So a concussion is a complex pathophysiological process, and it's actually considered a mild traumatic brain injury, and it can be a result of a direct or indirect blow to the head. So it could be, a ball hitting your head and then kind of shifting your brain, or it could be someone that's just really quickly jerking in a certain direction. And what occurs is the brain moves within the skull and hits against the cranium, and this causes swelling, which makes the brain extremely fragile and sensitive. And there are approximately 3.8 million concussions reported annually, and the highest incidences of these concussions occur in football, hockey, rugby, soccer, and basketball. But it's really important to note that there are many unreported cases. So it's very possible that the number, this 3.8 million, could actually be a lot higher, but we just don't know because they go unreported. Some of the symptoms of a concussion include loss of consciousness, headaches, dizziness, nausea, confusion, fatigue, sleep disturbances, balance and memory impairment, and light sensitivity. And then the actual molecular pathophysiology that's going on in the brain to cause these symptoms include alterations in the ionic balance, neurotransmitter activation, axonal integrity alterations, and then the alterations in the energy metabolism within the central nervous system. And it's really important to note as well that second impact syndrome is actually what causes the long-term damage. So you might experience these symptoms the first time a person has a concussion, but the second time they have that concussion, it's going to actually lead to permanent damage and symptoms that last for much longer than a few weeks. So next we're going to look at the actual prion protein that this article discusses. And a prion protein is a membrane-associated protein, and it's made up of 209 amino acids. These amino acids link together to form an alpha helix structure that can be seen right here. And it kind of looks like that DNA spiral that we see all the time in bio class, except it only has one helix instead of two. And this prion protein is highly distributed throughout the central nervous system, and it's associated with cellular adhesion, cell signaling, ion homeostasis, and neuronal protection. So it's a really important protein within our nervous system. And then our authors are suggesting that during a concussive event, the prion protein, which is membrane-bound, is then dislodged from its associated membrane and begins to collect within the systemic circulation. And this is a part of the cardiovascular system, so they are suggesting that if a person takes a blood sample, they will be able to measure the levels of the prion protein and thus see if a concussion has occurred. So it becomes a biomarker. And there hasn't been extensive research on prion proteins as a biomarker for a concussion, but they did find that the PRNP gene, which produces prion proteins, was upregulated after a huge blast or a big concussive event. So that's a really important indicator that prion proteins will be excessively produced within the blood after a concussive event. And then there are other protein biomarkers that have been used to identify traumatic brain injury, and these include S100B, Cleveland tau, or cleve tau, glial fibrillary acidic protein, or GFAP, and neuron-specific enolase. So we'll talk a little bit about GFAP later on in the presentation, but those are some important things to remember about prion proteins.
All right, so getting into the participants of the study, the participants were taken from a university in Canada, and they got participants from high contact sports such as football, soccer, wrestling, and also from low contact sports, which were generally said to be volleyball or cross country. And they also needed a control for this study, so they got participants who did not participate in sports as kind of the control. And so all these participants had to take a questionnaire that essentially asked them about their health and if they had any prior existing health conditions, and also if they had had a head injury in the past 6 months, and if any of the participants had a prior health condition or had a head injury in the past 6 months, they were excluded from the study. And so they used the sports concussion assessment tool in order to find participants that had a concussion, and so they were able to find 6, and this is simply just a set of 8 different sections that take you through physical and cognitive assessments that essentially are able to define if you have a concussion or not. And they were able to identify 6 of the athletes who did have a concussion. So for the first method that they used, they did a plasma separation. They took a small sample of blood and centrifuged it for about 10 minutes, and that was in order to promote the plasma isolation of the blood. And it was then, after it was aliquoted, it was stored for future analysis.
And then for the next method, they used an Eliza assay, which is an enzyme-linked immunosorbent assay, and it's used to measure analyte concentration. So this analyte that we're measuring is, again, this prion protein within the plasma. And they use recombinant prion protein to produce serial dilutions, and then a colorimetric reaction proportional to the concentration of prion protein. And that can be seen here with the various colors, and those colors indicate how much prion protein is present within the solution. And then they measured the absorbance of these and eventually did a calibration curve. And then next they focused on Western blotting, and this measured the protein concentration of plasma, and they first transferred the protein to a PVDF membrane at 1,000 volts per hour, and then the membrane was blocked, and then they used immunoblotting targeting to identify prion proteins, GFAP, and actin using primary antibodies. And then the protein bands were eventually analyzed and then normalized. And then finally, for the statistical analysis, the authors used both a t-test and a one-way ANOVA to determine the various ratios of prion proteins within our different groups. And first, they used a t-test to measure between male versus female, and then athletes versus non-athletes, and then the post-traumatic brain injury versus the baseline of all participants. And then they used a one-way ANOVA to determine whether or not there was significant variation of the prion protein between all the different age groups.
All right, so getting a little bit into the results, they kind of started with this question, whether or not plasma levels raise after a minor traumatic brain injury. And so they wanted to first explore if there were any variances between gender. So they wanted to compare males and females to see if plasma was higher or lower in one or the other. So after doing a t-test measuring the plasma levels, they found that there was no significant difference between the plasma levels in males versus females, which is good going forward to be able to test. So they then found a little bit different results. They found a slight significant difference between the off-season athletes and the control group non-athletic participants. And then the last part they saw, they wanted to see if there was any variance between different ages. And so they split up the participants into 5 different age groups. And after doing an ANOVA for those participants, they again found that there was no significant difference between any of the ages. So these figures kind of help explain this a little more. Right here is where you see the significant difference. What's interesting is that the normal or control participants actually have higher plasma levels, and it's something interesting that we'll talk about later, but that's just important to note. And then over in figure 3, you see the no significant difference between any of the age groups after measuring for variation of the plasma levels.
So next they looked at the prion protein within the concussed athletes. So during this study, they had 6 athletes that became concussed during their season. And so within 24 hours after they were concussed, they evaluated the initial signs and symptoms using cognitive testing for those participants. And then they had self-reported symptoms. So that helped kind of gather all the data about what they were experiencing and if they truly had a concussion. And then within that next week, they took blood samples from these participants so that they could measure the prion protein levels within those participants. And they found that the plasma prion levels were significantly higher than the baseline measures from all of the off-season athletes and the baseline measures from the entire population, including non-athletes. And then they had a t-test to compare just the people that had experienced concussion, so the before and after of the concussive event, and they actually found no significant difference between those two groups, which again we're going to talk about later in the discussion as to why that might be. And then lastly, they had immunoblotting that compared post-traumatic brain injury participants to the baseline of non-athletic controls, and there was again a significant increase in the prion protein, and also importantly a GFAP marker, which I talked about before as another biomarker for concussion. So just looking at these figures, it shows the significance between these 6 participants after the traumatic brain injury, and then the baseline of all the athletes, and then the baseline of the combined non-athletes and athletes. And then over here it just shows the difference between the individuals that at their baseline and then after their traumatic brain injury. So you can see that the levels of prion protein actually do increase, but not significantly enough to be considered a significant difference.
So getting into the discussion about this article a little bit and some of the importance with it, despite sport-related concussions being one of the most common of minor traumatic brain injuries, most of them are still unreported and even some are ignored. And this can be a really big problem with the second impact syndrome, which is why it's so important to have a reliable and easy way to measure if these athletes do indeed have concussions, so that there can be preventions taken place so that they don't get another one. And so in this study, it was really important that they did see a significant increase in PRPC after having a concussion, because that helps us know that this could be a potential biomarker for these sport-related concussions, like I said, that are so prevalent today. So focusing on the PRPC discussion in a little more specific, since PRPC does have these pathological ways that it helps, one of those is its neuroprotective role that it plays in the brain. And so the researchers hypothesized that in a concussive event, due to the blast that happens when a participant would get a concussion, the PRPC could be knocked off its lipid raft, and essentially then could collect within the cerebrospinal fluid and the systemic circulation. And so because it's most prominent in the central nervous system, it is possible that it could cross the blood-brain barrier in a bidirectional manner, and because of this disruption, it could allow for even more pooling of PRPC, which essentially means that there is a biomarker for a concussion, just that there's more of this protein. And so this protein has been previously used to identify other pathological conditions such as stroke, neuroinflammation, encephalitis, and such things like that. And so one of their challenges was to determine if there was a variance between age or gender, because in these previous biomarkers with other pathological conditions, they've seen it kind of a dependence on age and gender. So they wanted to make sure that there wasn't any for this test. And thankfully, they found that there was no significant difference, which makes it a really good testable hypothesis moving forward. And so, like I mentioned, they observed a lower protein level in the off-season athletes, and one of the things that they said could have contributed to that is because these athletes are in such good shape, they likely have a lower blood pressure than their counterpart controls. And so because this protein is often associated with the endothelial lining, people or participants with a higher blood pressure could be seen to have higher protein levels. And so we are able to say that elevated plasma levels are associated with the sport-related concussions, which is a really important thing from the study. And so for future research, they hypothesized that the central nervous system is contributable to plasma-soluble PRPC, which is really important going forward so we can study more closely the CNS and how it pertains to the protein. They also hypothesized that the severity of a concussion or of an injury could be contributable to the levels of protein found in the blood. For example, they found that a football player's plasma levels were higher than one of its counterparts. And lastly, an increased sample size, especially with concussed participants, is really important. They were only able to get pre- and post-test for 3 concussed individuals, so they didn't have a lot to go off of. But the fact that they did find these significant results with just those 3 participants is very important. But going forward, having an increased sample size is going to be very beneficial for future work.
So the ultimate takeaway from this article is that plasma-soluble prion protein does have the potential to be a useful biomarker for identifying concussive athletes that are at risk. Thanks so much.
Thank you. Thank you.